Hello friends, welcome to my YouTube channel, The Microbiology Digest. Today we will be discussing the second part of the structure of bacteria. In continuation to our last discussion, we'll be discussing the rest of the parts or components of bacteria after cell wall. So to uh, brief our previous class, we have discussed about the various external structures of bacteria. For example, glycocalyx or capsule, flagella, fimbriae or pili. Then cell envelope structures. Two cell envelope structures are there. One is cell wall, another one is cell membrane. So we have discussed up to cell wall. So today we will be discussing from the cell membrane and the other structures which are present in a bacterial cell. So this is a typical diagram. In our last class also we have discussed about this diagram. So directly we will start from the cell membrane, which is also called as cytoplasmic membrane in bacteria. So this cytoplasmic membrane, it lies next to the cell wall internally, and approximately it is around 7.5 nanometer thick. So it is a flexible structure composed mostly of proteins and phospholipids. So phospholipids, it constitutes around 20 to 30% of the structure, and the protein part, it constitutes around 60 to 70%. So these phospholipids, it forms a bilayer, and where the most of the proteins are embedded. These embedded proteins which are present in the phospholipid bilayer, they are called as integral proteins. And these type of integral proteins which are firmly embedded in the phospholipid bilayer, so these proteins can only be removed after destruction of the cell membrane. Then one more type of proteins are present which are called as peripheral proteins, and they are comparatively very loosely attached to the cell membrane, and they can be removed very easily with mild treatments like osmotic shock. So this cell membrane or cytoplasmic membrane in bacteria, in most cases they lack sterol. They do not contain any sterol, but some organisms, they may have some sterol like molecules which are called as hopanoids. So these hopanoids, they enhance the membrane stability. The inner and outer faces of the cytoplasmic membrane are hydrophilic, while the interior part is the hydrophobic. Thereby it forms a barrier to most of the hydrophilic molecules. Now, what are the functions? So, cell membrane, it performs some important functions. So, for example, it provides the selective permeability, means it acts as a selective barrier, and thereby it controls the substances which can or which cannot pass into the cell. Then second is the transport. Mostly it facilitates the movement of nutrients and the waste products. Then third is the energy transduction, fourth is the active transport, and last is the cell integrity. These are the important functions of the cell membrane. The next component in case of bacteria is the cytoplasm. So cytoplasm, it is the uh, part of the cell, bacterial cells, which lies inside the cytoplasmic membrane. Means cytoplasm is covered or enclosed by the plasma membrane or cytoplasmic membrane. So these are somewhat jelly-like structure which contains the internal components of a bacterial cells, and these internal components or structures which may be present, which are present in cytoplasm are ribosomes, chromosomes, plasmids, and other components which are required for bacterial metabolism. So as I said, this is essentially an aqueous fluid containing all the nuclear material and the other components, as we have uh, already discussed, primarily the ribosomes, chromosomes and plasmids. So these are present in the cytoplasm, in the aqueous fluid, where these components are present inside the plasma membrane. So this cytoplasm is the major site. It is the site for essential cellular functions. So most of the cellular functions takes place in the cytoplasm only. Cellular functions includes the cellular growth, metabolism, replication. All these important functions are it is carried out by the cytoplasm. Okay, so these are important functions for the bacterial cell, which are uh, carried out by the bacterial cytoplasm. So cytoplasm, it is very much essential to understand. It has different components. First component is the cytosol. So it is a watery portion. Okay, it is a watery portion, and it is composed of mostly 80% of its components are mostly uh, water. So it is the cytosol. So next component is the nucleoid. So nucleoid, it is a chromatin dense region containing the bacterial chromosome, DNA, proteins, etc. So they are present in the nucleosome. Nucleoid. Next is the ribosomes. As we know that ribosomes, they are responsible for the protein synthesis, or they are sites of the protein synthesis. So they are also a component which is present in the bacterial cytoplasm. So next is the inclusion bodies. These are storage structures. There are different types of inclusion bodies are there. So they are present in the cytoplasm only. Then the plasmids. Plasmid, as we know, these are small circular bacterial DNA. Okay, these are different from the bacterial DNA which is present in the nucleoid. So, but they are small circular extra chromosomal DNA. Okay, they are also present in the uh, cytoplasm only. Now, coming to the functions of cytoplasm, as we have already discussed, metabolism is an important function which is carried out by the cytoplasm. Then cell growth and replication. This is also for survivability of the bacteria. Cell growth and replication, it takes place in the cytoplasm. Then intracellular organization. This is also an important function carried out by the bacterial cytoplasm. Now next component is the nucleoid. So this is again already we have discussed, this is present in the cytoplasm. So this is a irregularly separate region containing the chromosome and the numerous proteins. Means bacterial nucleoid, this is not this is the nuclear material actually uh, containing the bacterial DNA or chromosome as well as the proteins. And there is no this structure is not a membrane bound structure, so that is why it is called as nucleoid, not nucleus. Okay, because this is not a membrane bound structure in case of bacteria. Okay, so bacterial genome, it is composed of a single haploid circular chromosome containing a double stranded DNA. So this is the genome structure, and small amount of proteins, RNA, these are also associated with the nucleoid. Okay, so bacterial genomes, they vary. Their size it varies from bacteria to bacteria, species to species, and because it is because of its length, it is extremely folded structure. Chromosome in case of bacteria, it is extremely folded structure, and this is can be this nuclear material in case of bacteria can be demonstrated by using light microscopy, but for that we need to use a special stain which is called as Fuelgen staining method. So this is a staining method for demonstration of DNA, specific DNA by light microscopy. Okay. Next structure present in bacteria is the, or it is a component of the bacterial cytoplasm. They are plasmids. So plasmids are, as already we have discussed, these are small circular double-stranded DNA, and they are extra chromosomal. They are not a component of each of the bacterial chromosome. So that is why they are said as extra chromosomal small circular DNA, and they are independently they can go for independent replication. Their replication is not dependent on the bacterial chromosome. So that is why they are extra chromosomal small circular DNA capable of independent replication. This is the definition of plasmid. So these plasmids, they are responsible for providing antibiotic resistance, or they takes part in the bacterial conjugation, because they helps in transfer of genetic material from one bacteria to another bacteria. Okay, and these plasmids, they are also used very commonly in the experiments using recombinant DNA technology. So these plasmids, they are one of the very important component which are very widely used for recombinant DNA technology based research. So there are different types of plasmids in case of bacteria. First type is called as conjugative plasmid. From the name itself, you can understand what is the function, what may be the function of conjugative plasmid. Then second type is the resistance plasmid. This is responsible for the providing antibiotic resistance or conferring antibiotic resistance to the organism. Then cold plasmids. So these cold plasmids, they contain the genes which are required for synthesis of some bacteriocins, for example colicin uh, by Escherichia coli. Then virulence plasmid, they encode the factors which makes a bacteria pathogenic. Okay, these are virulence plasmid. Then metabolic plasmids, these are different type of plasmids which are present in case of bacteria. So five different types of plasmids we have discussed. One is conjugative plasmid. It is responsible for transfer of bacterial genome from one bacteria to another during conjugation process. Then resistance plasmid, this is responsible for conferring antibiotic resistance. Then cold plasmids, these are responsible for coding the genes for synthesis of some bacteriocins. Then virulence plasmid, related which confer the virulence factors to the organism, and finally the metabolic plasmids. So next component is the bacterial ribosomes. So ribosomes, as we all know, they serve as a machinery for the protein synthesis. So in case of bacteria, the type of ribosome present is 70S, and this 70S ribosome is composed of two different subunits. One is smaller subunit, another one is larger subunit. The smaller subunit, it is the 30S subunit, and the larger subunit is the 50S subunit. This subunits, they act as a site for action of antibiotics. For example, streptomycin. This streptomycin antibiotic, it binds with the 70S ribosome of the bacteria, and it blocks the protein synthesis, and thereby it can kill the organism. So there are many other antibiotics are there which acts through the binding with the different subunits of bacterial ribosomes. So next component is the mesosomes. So mesosomes actually they are the invaginations of the plasma membrane. They are mostly encountered in case of gram positive bacteria, and these mesosomes, they are formed by the folding of the cytoplasmic membrane, and so they appear as vesicular structures, or sometimes they may appear as a tubular structures. Okay, so there are different no clear-cut evidence about the functions of mesosomes. Still, different studies are going on. But historically, initially these mesosomes, it was thought that mesosomes may be associated with some important functions like respiration, then DNA replication, and they may serve some other roles. But present findings, they have shown that these functions are not associated with mesosomes, but they are formed as some artifacts during the uh, uh, processes of the electron microscopic procedure. So when we go for electron microscopic examination, so when we prepare the sample or the organism, so during that process, these mesosomes, they appear as some artifacts. But yet the exact function performed by the mesosomes are still not clear. Uh, Studies are new evidences are being put forwarded by different group of workers, but no conclusive report has yet been confirmed regarding the proper function provided by the mesosomes. Then next components are the which are present in the cytoplasm. They are storage granules. So from the name itself, you can understand what may be the function of these storage granules. So there are different types of storage granules are present in bacteria, which may be present in the cytoplasm. For example, glycogen inclusions. So these storage granules, they are present. They are also called as inclusions. So glycogen inclusions, then sulfur globules or inclusions. So they may be, or polyphosphate granules, they may be present in different form, but they are mostly formed by the aggregation of the substances that may either be organic or inorganic. Okay, so these substances, uh, they are densely compact structure, and they do not generally have a membrane covering. Means they are densely compact structures, and they do not, and without a membrane covering. And mostly these storage granules, they are thought to be the reserves, or they can store their nutrients for their time when they will the bacteria will face some unfavorable situation. So these are uh, we can say these are the storages of the nutrients when bacteria will face some unfavorable situation. These storage granules can be used as a nutrient by the organism. So these are uh, nutrient storages by bacteria. This inclusion or storage granules are nothing but these are nutrient reserves of the bacteria for the unfavorable situation. Okay, so different types of inclusions may be there: glycogen inclusions, sulfur inclusions, polyphosphate inclusions. So these inclusions may be present in different depending on the species of the bacteria. The inclusion may also be different. So this is all about the bacterial structure. uh, I tried to cover each of the component in a very simple way. So I hope this information will be useful for you, and try to understand the component or the structure and the functions performed by the respective structure. And most of the structures in case of bacteria, they are mostly related to the survivability of the organism. So that is why it is very much essential to understand the functions of each of the components or bacterial structures. So I hope this information will be helpful for you. If you find it is helpful, please subscribe to my channel and give your comments so that I can further add the missing information. If anything I may miss, then that I can incorporate it in my future discussions. So with this, thank you very much. Thank you.